I'm going to introduce Simon and his work. I'll take questions and answers later, and I'll moderate the questions and answers that come through on the Q&A that you can see at the bottom of your Zoom options. I won't be opening the mic to any of the participants. It's just to make sure that we stay fair to everybody that's participating. So thank you so much, Simon, for making the time and being interested to present. We're really excited to hear what you have to say about unique effector fold and insights into the KEX2 protease processing revealed by the crystal structure of SN-TOX3 from the necrotrophic fungus Parastagonospora nodorum, and that is a tongue twister. So I'm looking forward to hearing what you have to say. Thanks so much, Simon. Over to you.
No worries, Lisa and Adam. Thank you. So I'm just going to, and also thanks to OPP for giving me the opportunity to come and talk a little about some recent work that we've done in the lab. Also, we've been working on this though for some time, so just go ahead here and share my screen. We could get started. So yeah, awfully long title. Sorry about that, Lisa. But today what I'm going to talk about is our work in solving the crystal structure of TOX3 from Parastagonospora nodorum. I'm going to talk a little bit about the structure, but I also want to talk about some of the insights that we've sort of found whilst working on TOX3, and particularly talk about KEX2 protease processing of fungal effectors. So I recently started my own research lab at the Research School of Biology at the ANU, and my lab is predominantly interested in effectors and receptors. And when I say effectors, I refer to we're mostly interested in fungal effectors. So these are proteins that fungi produce and utilize to aid in their infection strategy of a plant cell. And we're also really interested in recognition at the plant level by disease resistance proteins, either resistance proteins that are known to survey the apoplast and interact with the apoplastic effectors, and also the NLR resistance proteins, the intracellular resistance proteins. But for my talk today, what I really am going to focus on is effectors. So effectors, as a lot of you would know, are often small secreted proteins. A lot of them have cysteine rich, and this is involved in disulfide bonding. And also what's familiar to a lot of people as well is that when we look at the sequence of effectors, they very rarely provide any information about function, because we really often don't see any sort of homology to proteins with known function or recognizable motifs. And so what we try and do to sort of combat this is to utilize structural biology and protein biochemistry approaches to understand effector function. So because of the techniques we use, we can look at a lot of different pathosystems. And so these are predominantly the pathosystems that we work with. And we can't do this without really strong and important collaborations that I have both in Australia and internationally. So the stories today are going to predominantly focus on these necrotrophic fungi, and the one in particular I'm going to talk about today is Parastagonospora nodorum, and this is work that I've been doing with Peter Solomon. Also going to touch on some work that I'm doing with Professor David Jones, also at ANU, on Fusarium wilt. We do also a lot of work on flax rust and stem rust effectors, but I'm not going to talk about these today. So the outline of my talk, I'll be talking about producing cysteine-rich disulfide bonded effectors. This is not a trivial task, and what I really want to do is provide, I guess, some insight into how to do this and some tips and tricks. I'll also talk about the TOX3 structure, and then applying some of the lessons we learned while solving the TOX3 structure to other cysteine-rich effectors, and then how this has come towards us defining the K2PP effectors, and I'll get onto this towards the end of my talk. So TOX3 is I think a pretty awesome effector protein in that when it's produced and secreted by Parastagonospora nodorum, and in sensitive wheat lines, so wheat lines that carry an SNN3 gene, it actually induces cell death. And this cell death is really important for this necrotrophic pathogen because it enables the pathogen it's a strategy of the pathogen to cause necrosis. In the absence of SNN3, you don't get any necrosis, and of course in the absence of the effector you don't get necrosis. But what's also really interesting is this phenotype here is from the protein alone. So it's causing this cell death response that is completely dependent on the presence of this susceptibility gene. So TOX3 was originally identified and cloned from Tim Friesian's lab, and it's a secreted protein, 230 amino acids, and it has this putative prodomain. And this is important. We'll talk about this throughout my talk. And it also is defined by 6 cysteines, which are believed to form 3 disulfide bonds. And Tim's group actually originally identified this from culture filtrates of Parastagonospora nodorum. And what's important is that the protein itself is sensitive to a reducing agent. So this is a titration they did here where they used dithylthreotol, which is a reducing agent which reduces disulfide bonds, and they showed that by incubating the protein in DTT that it actually lost the function. So we know that the disulfide bonding is important for the function of TOX3. But when you look at the sequence, there's really no clues about what the protein might look like or how it might function. So I go a long way back. This journey really started back in 2011. I was a postdoc in Boston Kobe's lab, and we were predominantly working on flax, rust effectors, and also NLR proteins. And I happened to bump into Peter Solomon at a bar in far north Queensland, Cairns, at a conference. And Peter had just given a talk about TOX3, and I was really interested in sort of understanding more about this protein. And so a few years later, I sort of established a collaboration with Peter, which is actually continues now to this day. And we actually have labs next door to each other. So this was a slow process. So the first thing I needed to do was try and produce these cysteine-rich effectors. And what I looked to try and do was to do this in E. coli. There had been some reports of producing TOX3 in Pichia pastoris, but the yields were really low. And so we wanted to, in fact, you couldn't really see this on a Comasi stain gel. So we wanted to try and do this in E. coli. We tried a lot of things, but sort of settled on this E. coli strain produced by NEB known as Shuffle. So Shuffle was designed in a way to promote disulfide bonding in the cytoplasm of the E. coli cells, and so it has a number of proteins or genes knocked out, which sort of promote the reduced environment of the cytosol in E. coli. So these are knocked out, but it also overexpresses a protein called DSBC in the cytosol. And this is actually really important and actually involved in shuffling of disulfide bonds and correctly pairing of disulfide bonds. So we started to use Shuffle and we had some success. So we were able, so this is an SDS page gel of TOX3. What we could do, we could produce it and purify it from Shuffle. And then what we're doing here is we're just injecting the protein with a needleless syringe into a wheat leaf. And in this case, we're looking at Corac, and so this is an SNN3 plus line, and then here we have an SNN3 minus line. I should have had that written in there. And so we're seeing this necrosis because of, well, this cell death because of due to the protein. And so we actually, this work was published, and it talks about a number of different cysteine-rich effectors that were produced in shuffle when shown to be functional. But we had a problem at the end of this, and the main issue was yield, and we had a still really low yield. So we were getting about 0.05 to 0.1 mgs of protein per litre of E. coli culture. And we need a couple of mgs at least to sort of start crystallization. So I was fortunate, so I sort of chipped away at this. I was doing a lot of other things, and I chipped away at this. I was fortunate that Bostian Kobe, who was my boss at the time, let me do this. But I was also incredibly fortunate to have Megan Ottram join the lab to do her PhD, and everyone in the lab was working on tear domains, and somehow convinced Megan to actually take up the challenge of TOX3. And this was, I'm not sure if she had a time over she would have done this, to be honest, but this was a really tough project. So this is back sort of mid-2015. And Megan sort of walked into the lab, and I was still with Bostian, but I was sort of her main supervisor, I guess. And so I came up with a really, what turned out to be a terrible idea. And that was that if we can make a little bit of TOX3 using shuffle E. coli, then Megan should just do 20, maybe even 40 litres, and we would have enough for crystallization. And I have to say, this experiment failed miserably. And I was lucky that Megan didn't leave, so she hung around. And what ended up, we tried a lot of things, but the key, so some of these was we tried different expression strains. So there's other strains of E. coli that we looked at. Origami was one of these. It didn't work from us. We tried yeast. That didn't work at all. Didn't work from us either. We also tried different induction temperatures, all sorts of things. We worked on this for a long, long time. But in the end, what was really important was the addition of a fusion or solubility partner at the N-terminus. So instead of just having a his tag, we also had a fusion partner. We initially started by using Sumo, and I'd like to actually just thank Mark Banfield's lab for making the suggestion of actually trying GB1. Because GB1, as you can see here, so this is a size exclusion chromatography run of TOX3 that's been cleaved away from these fusion partners. One of them wasn't a fusion, it was just a his tag, and then we have the Sumo and GB. And we saw, so the tags being cleaved away, and we just have TOX3 left. But what we saw doing this, we got a massive enrichment using GB1 as our fusion partner. So it was almost 10 to 20 times increase in solubility doing this consistently. And we've also seen this for other cysteine-rich effectors, and I'll get onto this in a minute. So that was great, because what that meant was it opened up the door for crystallization. And it was a really exciting day when we got these TOX3 crystals. They were tiny needle-like crystals, you can see that here. And this was great, but what sort of happened was we couldn't really optimize these, and they diffracted X-rays poorly. So we saw 9 angstrom diffraction. And so we thought, so what's going on here? And what Megan did was she actually picked the crystals out of the drop, and she had a look at them on a gel. And what we were seeing, so before crystallization, we sort of had a full-length TOX3, and what we were actually seeing is after crystallization in these drops, we were seeing that the protein actually had migrated further down our STS page gel. And when we sort of worked this out, we did use mass spec techniques and N-terminal sequencing techniques, and we actually identified that this N-terminal region of TOX3 was being cleaved off actually in the crystal drop, and it was this part of the protein, the N-terminal part of the protein, that was forming crystals. And when we sort of, so we wanted to look at this a little further, and we wanted to look into to see what happens in Stago. And so I should say Parastagonospora and nodorum. We call it Stago in the lab. So we grew up some P. nodorum in liquid cultures. So this is SN15 wild type, and we had a tox3 knockout. And then we looked at what was getting secreted, and we actually had antibodies against tox3. And what we could see was that the protein that we were picking up was actually tox3 without this predicted prodomain. And so when we actually had a close look at this, and also we went back and looked at Tim's the paper that came out from Tim Friesen's lab about this, they actually talked about this putative KEX2 cleavage site. And so, and this was exactly where we were seeing the protein being degraded that formed crystals. And so you might think, you idiot, you should probably read Tim's paper a little better. But to be honest, when we first started trying this, I actually made a construct that started from this, and we started from this tyrosine here, and we tried to make this, and we couldn't produce it at all. We couldn't get any soluble protein. Nevertheless, we thought we'd try again, and we tried with our GB fusion. And Megan was quite upset here because we thought that this could work quite quickly and quite easily, but we went back to getting essentially no protein. So this was sadness, and it turned into also into some frustration. So we had to come up with another method, and what we ended up doing was we actually just produced the full-length protein as we were doing before, and then we exposed it to KEX2 in a tube. So we just took some purified KEX2 protease, and what Megan was happy again because we had our yields back, and we could actually make tox3 KEX2 take off this M-terminal domain. And this was really the most important thing in solving the tox3 structure. So here's some, I like showing crystals because I think they're beautiful. This is a plate-like crystal of tox3. This is the structure. For any crystallographers out there, we used halide soak to solve this using bromide. And so this is the structure here. What we have is an 8-stranded beta barrel structure. The protein is tethered sort of back to itself by these 3 disulfide bonds that are indicated here in yellow. And so we had really nice resolution of this. The details of this is actually, we just dropped on to bioarchive as well. So fantastic. This took a long time, but we got here, and here was our structure. So the thing you do when you get a new structure is you want to try and work out, based on other structures, what it might do. So we took the coordinates and we threw them against the protein data bank and to see what kind of structural homology might be out there. So we used the Dali server. And I think excitingly, initially, what came back was that we had similarity with bacterial pore-forming proteins and toxins. And so what these pore-forming toxins do is they actually oligomerize on the surface of the membrane. They punch a hole in the membrane, and then then the cytosol comes out and they kill the cell. And so the top list of effectors that had homology were all from this class. But the actual structural similarity really only was found in the cap domain of these proteins. And tox3 didn't sort of have this rim and stem domain that we see in bacterial forming toxins. And so this is actually the oligomer structure of bacterial forming toxin, and we have tox3 superimposed on. And what we sort of even realized just straight away from this structure is tox3 doesn't have the protein bits to sort of form a pore. So it would need to associate with other proteins in order to form a pore. And that's where we're sort of sitting with this. We haven't really been able to take this further. We've tried some different things. We haven't seen any suggestion at this stage that tox3 is functioning at all in this way. What we did do is we used the structure to look at some of the interactions that tox3 has with pathogenesis related protein 1. I'm not going to go into this because actually Peter Solomon gave a talk about this probably a month or two ago on OPP, and so that all of that work is actually already up on YouTube, so you can check that out if you like, if you missed it. So what I want to talk, just for the remaining of the talk, is about tox3, and is a KEX2 process prodomain. So we're calling these K2PP effector. So what is KEX2? So KEX2, it actually sits in the late Golgi, and what it does is it processes proteins during secretion from yeast, and fungi have a KEX2 homologue as well. So they're calcium dependent, and what they do is they recognize this lysine-arginine or arginine-arginine double basic motif, and they cleave at this motif and they take off prodomains, and so then you get the mature form of the protein being secreted. So, you know, we were sort of working all this out with TOX3, and at the same time we're working on a number of other effectors. And so some of these were from Fusarium wilt, and then another effector from Stago, known as TOX-A. So when Martin Rep identified these effectors from Fusarium wilt, so FOL6-1 or AVR3, also known as or FOL6-4 and AVR1, they actually identified this KR or this KEX motif and suggested that these proteins are cleaved by KEX during secretion. So that was already known. But so another thing I just want to mention, so we're working on these proteins, and this is just an example for anyone who wants to do a little bit of protein biochemistry and work on effectors. Use a fusion protein for these, right? So both SUMO and GB dramatically increase the amount of protein, the yield of protein that we can get with all of these different cysteine-rich effectors that we're working on. Anyway, back to sort of this idea of KEX2. So we could, so it had already been suggested that these proteins here would be processed by KEX2 from the literature. And so what we did was we actually could produce all of these proteins. We exposed them to KEX2. So this is in vitro. So we took some purified KEX2. You can see that we get the N-terminal region cleaved off in all of these. You also can sometimes get multiple bands, and this is really comes about because there's, you know, in some of these, there's actually in FOL6-1 and also in FOL6-4, there's an additional putative KEX cleavage site. And also, you know, you sometimes get these dibasics coming up, you know, in other regions of the sequence. But I think what was, I guess, interesting to us and a little bit of a surprise to us was that FOL6-6, which has an SR region, and also TOX-A, which has a PR, was also cleaved by KEX. And when we started to look at more effectors, what we sort of started to notice was this LXXR type motif at the N-terminus of a whole heap of different secreted effectors. And so we sort of, I guess, a lot of the effectors that it had been identified, it was either a KR or an RR had sort of been hypothesized to be cleaved by KEX. But also when you look at the literature, we know that KEX can have a different preference for the P2 site. So there's a paper in PNAS quite a long time ago, published a long time ago, and it sort of showed this preference for different sort of amino acids at this P2 position. And also there's a more recent paper where they talked about the importance of the P4 position in KEX cleavage. And when we looked at sort of the work that what we'd seen for the with the effectors that we'd worked on and the information from these, we sort of then started to hone in on this LXXR. So we're also really lucky at ANU to have Jana Spersnader in just across in another building, and so I was talking to Jana about this, and she said, oh, she could help out. So Jana did a search for KRRR and this LXXR in her effector set from effector P 2.0. And what she also included in this was, so she sort of a criteria where we're looking for that the N-terminal sequence before the motif was less structured than after the motif. And this was really because our observations with the other effectors was that we saw these regions, or these prodomains, were almost void of secondary structure. And in doing this, she picked up 28% of the effectors in the data set actually had a KEX2, what were putative K2PP effectors. So this was a lot higher than, I guess, sort of we had thought, and I'm thinking maybe the community had thought. So we think that KEX2 processed prodomain effectors are highly prevalent in fungal effectors. And I guess what I want to do is, in the last couple of slides, is just touch on the implications and considerations for people that might be working on K2PP effectors. So the first is, if you're looking to do any sort of protein biochemistry or structural biology on this, then we have some things that we found that I think would be important. One of those is that, again, and I'll just highlight here, so this is work done by now a PhD student in my lab, Daniel Yu. And so this is on the FOL66 effector. And Daniel showed, like with TOX3, we could get a lot higher yields when we produced FOL66 with a GB fusion. We also find that if we take the prodomain off, right, altogether, and then we just try and make it with a GB fusion, we get no protein whatsoever. So at least in a heterologous system, these prodomains are really important for folding of the effector. So this here is essentially just showing that we're basically getting no protein at all when we take off the prodomain. The other thing is, if you're going to do some crystallization, taking off the prodomain seems to be pretty important. So this is some Daniel showed when we set up the full-length protein compared to when we actually took the prodomain off. And so Daniel has some crystals here. Now, unfortunately, these don't diffract X-rays at the moment, so we're still improving this and hoping to get a FOL66 structure soon. I think the other thing to consider is when you're working on these effectors in plantar. And I know that there's a lot of groups that do a lot of their effector studies in plants. So plants don't have KEX2, and they, I guess what we say, they may not process the prodomain. So I showed before this data where we know that TOX3, when it's secreted from Stago, that prodomain is removed. So not so when we do it in benth. So we take, in this case, we've got TOX3 in benth. We've got this, we're using here a GFP fusion, and we're probing with GFP. But what you can see is we're actually secreting from benth full length. So this is secreted TOX3, and we're secreting full length TOX3 with the prodomain intact. So you might think, oh, well, that may not matter. I mean, is the prodomain really going to be an issue? Well, it can be. So, and this is our example. So if we take TOX3 and we infiltrate TOX3, full-length TOX3 with the intact prodomain into susceptible wheat lines, we see this necrotic response. In the absence of the prodomain, so when the prodomain is taken off, we see a lot stronger necrotic response, suggesting that in this case, the prodomain is really preventing TOX3 from actually from causing that cell death and causing that necrosis in susceptible wheat. Importantly, KEX2 does nothing when you infiltrate that. So I think some conclusions and broad implications. I think that one of the first things is be careful what you promise at the conference bar. This has been a long time coming. We've worked on all this stuff for a long time, but I think we've made some big inroads here, and Megan finally got that TOX3 structure before her PhD, which was fantastic. I'm fortunate to have Megan now working as a postdoc with me. I hope I've provided some hints and tricks to producing cysteine-rich effectors. There's more information here in our preprint that you can go and have a look at. I think that the LXXR motif, we see that we observe this in N-terminus of, I think, more fungal effectors than in many fungal effectors, right? And I think that K2PP effectors therefore might be a lot more prominent than we thought. The presence absence of the prodomain, it can, at least with TOX3, can affect function. So I think this needs to be taken into consideration when you're doing functional studies with your favourite effector. I want to just make note that LXXR is not an RXLR, and I think this is really important just to mention. So the KEX2 cleavage site is common in fungal secreted proteins, and it doesn't necessarily mean that they're effectors. So a lot of proteins are cleaved by KEX during secretion. So it's not really a diagnostic for an effector. And one thing that we're really interested in is understanding the role of prodomains and what these might be doing in terms of effector folding and how they aid in secretion. So I'll finish there. Thanks everyone for tuning in this morning, this afternoon, or this evening, depending on where you are around the world. I'd also particularly like to thank Megan. She's done a huge amount of work on this. Incredibly resilient, I think, because there's a lot of times where I think a lot of people would have just given up on this, but she got there in the end, and we've got some real insight from that. Peter and the Solomon lab work very closely with those guys, as well as the Jones lab. Also, Jana's been fantastic. Boston, we still collaborate with Boston. He was a fantastic boss when I was there. I have great collaborations with Daniel Eriksson. He's at the Australian Synchrotron, and he helps us out with all of our structural stuff. I work in a really awesome environment in RSB Plant Science with colleagues like Peter, David, John Rathjen, Uli Matissas, and Benjamin Swessinger. And also thanks to Mark Yoles, Adam Bentham, and Mark Banfield, because they actually provided us with some of the Golden Gate compatible vectors that we've sort of engineered further to do some of this work. So I think I'll stop there. I'll stop sharing my screen and I'll invite some questions.
Thank you so much, Simon, for the very interesting talk. I think let's open up for questions and then we'll go there. So we have no questions, have to run, but thanks. So just anyone else with questions, while we wait, I'm going to ask a question. Sure. So this is not my area of expertise at all. Maybe you can explain the implications of this for someone that doesn't have much experience. So when they watch the video, they can just get a bit of a backtrack on how this fits in the bigger picture, I want to say, and why it's important to consider the biochemistry associated with fungi or with pathogens and their host plants.
Yeah, so I think, I guess I kind of skip over that a little bit at the start, but I think what we really want to understand is what strategies are fungi using when they infect plants. And we know that they secrete a host and utilize a whole heap of effector proteins during infection, but we don't know a lot about how those effector proteins are functioning, what they're targeting. And so what our lab's really trying to do is try and pick this stuff apart and try and understand this. And so we hope we can sort of get help using structural biology techniques. I'm going to be honest, there's a lot of effector structures out there that haven't necessarily, we haven't got the structure and gone, oh, beauty, now we know the function. I think that there's a lot of people that will agree with me on that. But we actually, having said that, we actually have some recent structures where of fungal effectors where the structure is really helping us with the function. So I think with TOX3, I think it's important. I think the main thing that we got out of our work with TOX3, apart from the work that Peter presented a couple of weeks ago, was really this idea that I think there's a fair few fungal effectors out there that are being processed as they're being secreted. And I think you've got to look out for this, because I think that's going to have implications no matter if you want to do protein biochemistry or if you want to, wherever you want to study these effectors. So, I mean, we see with TOX3 that during secretion, it's treated differently by its host versus the plant. So I think this is really important.
Thanks, Simon. So, question, do you have an explanation for why TOX3 was cleaved at exactly the KEX to cleavage site in the crystals of full-length TOX3?
Yeah, I think this is, no, I don't. So I think there's certainly a protease, a host protease from bacteria that's doing this. So I think that was a contaminating protease that that ended up in the crystal drop. We think we think this because we actually did some further purification of TOX3, and we no longer got that cleavage. So we used a further chromatography step, and we no longer saw that prodomain being cleaved off. But no, I don't exactly know the E. coli protein that's responsible for cleaving. So KEX is sort of specific to yeast and fungi, but there are functional, and they're not structurally related, but there's functional sort of homologs in other organisms that will cleave at sort of these double aspartate or these double basic residues.
Another question, the 28% of the K2PP effectors that were higher than you thought, what are the implications for this
Yes.
for the community and for research?
So I think you should just, I guess what we're trying to say, so I think the first thing I would say is that these are putative K2PP effectors. You know, we haven't verified these. This has just come up from, you know, from Jana having a look put under these criteria. It could be more, but I think people should start looking for this, and I think not just focus on looking for like a double, just the two basic residues. I think we see, particularly at that, essentially that P2 position, you can have a lot of variation there. And I think that leucine at the P4 position is really important. So I would be looking for this sort of LXXR motif in your fungal effectors, at the N-terminus of your fungal effectors.
Great, thank you very much, Simon. So are there effectors that have unstructured prodomains that don't have KEX2 cleavage sites?
Good question, yes. So agar M, for instance, so flax rust effector, that has a really long N-terminus that doesn't seem to really have any structure. We don't know what that does either. We saw the structure of AVRM, but not that region, of course. So yes, there are. Yeah, and are they being cleaved by something else? I'm not sure.
Next question. Have you looked for any sequence homology within the prodomains of the various K2PP putative effectors that may suggest any functioning, for example, targeting or chaperone functions, etc.?
Yeah, we have, and we haven't picked anything up yet. We could probably revisit this. This is, I think there's a lot of stuff with chaperones that it's not really sequence specific. It's more, well, there's certainly a lot of chaperones that aren't really sequence specific. They're just more sort of, I guess, the properties of the amino acids, so hydrophobicity and this type of stuff. Nothing obvious.
All right. We'll take one last question. Let's have a look. All right. Okay, so I have a question relating to the structure, where you said there was only one part of the three, the cap, the rim, and the stem. Is this something that you expected, or?
So I guess when we were looking to to solve it, we didn't know what we were going to get. When we first got the structure and we sort of looked for structural homology, we got really excited initially because we got these hits. But then when you sit down and spend a little bit of time on it, you know, these bacterial pore-forming proteins really can't function without these other domains. And there are bacterial pore-forming proteins that interact with other proteins to form pores. So, yeah, I guess, yeah, in some ways, surprised, maybe disappointed that this wasn't. But clearly, I think the evidence we got so far do not suggest that TOX3 is a pore-forming protein.
Well, I must say congrats to you and your team, and is it Megan for finishing and persisting through on her PhD. Really congratulations to you all. Seems like it was a challenging project, but it seems also that it's been paying off. So thank you very much for sharing your tips and tricks and saving other people perhaps some of the pain that you've had to go through when if they pick up on what you've been doing. Yeah,
no worries at all. And thanks for having me, and thanks for people for listening. And yeah, feel free to email me any sort of further questions, or if anyone out there is going to embark on doing some of this stuff and doesn't have a huge amount of protein biochemistry experience, we certainly can offer some advice. So.
Great. Thanks, Simon. And just to wrap up, I'm going to send in the chat box virtual seminars. If you'd like to contribute, you're welcome to use the Google Docs link, which I see I've now shared, as well as the link to the Peter Solomon YouTube talk, the Slack channel, as well as the Twitter for Open Pathology. So thank you everybody for making the time to be with us this evening, especially for those of you that are in the Australian side of the timeline. I know it's getting late, so thank you so much for making the time, and we'll see you at our next OPP seminar. Thanks, Simon.
Thank you. Cheers, guys.